Hey everyone, and welcome to this explainer. Today we're looking at what might honestly be one of the most revolutionary biological breakthroughs in recent memory. If you've been following the evolution of gene editing at all, you know we've gotten incredibly good at snipping and fixing DNA. But what happens when we program a molecular machine not to edit, but to seek and destroy?
It really all starts with this massive, looming question. How do you drug the completely undruggable? For decades now, the persistence of certain tumor mutations has been the ultimate biological mystery. We're talking about the cancers that evade traditional therapies, the ones that build up resistance, and the ones driven by mutated proteins that our best medicines simply cannot latch onto. Well, today, we're going to look at how scientists are finally answering this question by literally turning a cancer cell's own genetic instructions against it. Okay, let's dive into this. Here is our roadmap for today. 1. The undruggable cancer problem. 2. Meet the chromatin shredder. 3. Targeting genetic fingerprints. 4. The p53 holy grail. And finally, 5. Shrinking tumors in vivo.
Section 1. The undruggable cancer problem. To truly appreciate this breakthrough, we first have to understand the incredibly high stakes that cancer researchers face every single day. We're anchoring our entire explainer today on some incredibly cutting-edge, peer-reviewed science. This is an accelerated article preview from the journal Nature, authored by an absolute powerhouse team of researchers, including Nobel laureate Jennifer Doudna's lab. And they've discovered a way to reprogram a CRISPR system to do something entirely unprecedented in mammalian cells. Now, to grasp why their discovery is such a game-changer, you really need to take in this staggering number. 40 to 50%. That represents the percentage of all human cancers driven by a mutation in just one specific gene, the TP53 gene. This gene produces the p53 protein, which normally acts as a tumor suppressor. It's the good guy. But when it mutates, it flips and it essentially drives the cancer. So, the crucial point is why traditional therapies completely fail against these mutant proteins. p53 is famously deemed undruggable. But why? Well, mutant p53 proteins lack the defined physical pockets that small molecule drugs need to bind to. It's like trying to put a key into a door with no keyhole. Furthermore, trying to chemically reactivate a defective transcription factor is biologically exhausting for the body. And if you try to broadly activate p53, you get massive, severe toxicities because you're triggering non-targeted cell responses. Standard medicine simply hits a brick wall right here.
Section 2. Meet the chromatin shredder. What if, instead of hitting that brick wall, we bypass the protein entirely and target the cell's underlying code? Enter CRISPR-Cas12a2. I want you to think of this not as your standard gene editor, but as a highly programmed cellular assassin. In nature, bacteria use this for something called abortive infection. That means if a virus infects a bacterium, the bacterium uses Cas12a2 to recognize the viral RNA and then basically self-destructs to save the rest of the bacterial colony. The brilliant leap here is that scientists have repurposed this RNA-guided nuclease. Now, when it recognizes a specific target RNA, it activates and begins cleaving chromatin, which is the tightly packed DNA in the nucleus, in what's called a trans manner, meaning once it's triggered, it just cuts surrounding DNA indiscriminately.
And this brilliantly illustrates the exact molecular mechanism at play. Step 1. Cas12a2 is armed with a guide RNA and hunts down the specific mutated cancer RNA transcript. Step 2. The exact moment it binds to that cancer RNA, the nucleus unleashes its transcleavage activity. Step 3. It literally begins shredding the eukaryotic chromatin, specifically prioritizing the linker regions between nucleosomes. Finally, Step 4. Overwhelmed by acute DNA damage, the cancer cell simply cannot divide. It undergoes what's known as mitotic catastrophe, and it dies. The absolute beauty of this system lies in this exact contrast. Think about it. In a healthy cell, the wild-type cell, the RNA trigger is completely absent, so Cas12a2 just sits there. It does absolutely nothing. No toxicity, no shredding. But in a cancer cell harboring the mutant RNA transcript, that mutant RNA acts as a tripwire. Cas12a2 binds, activates, and acts as a precise shredder, completely eliminating the mutant cell from the population. Section 3. Targeting genetic fingerprints. Knowing this works in theory is one thing, but how do we ensure absolute precision? Well, because it targets RNA, Cas12a2 is basically trained to hunt genetic fingerprints. Cancer cells are notoriously messy. They leave behind these cellular RNA signatures that normal cells just don't have.
This means Cas12a2 can be programmed to identify transcripts from overexpressed oncogenes, or even entirely unique junctions that get created by DNA deletions.
For example, the researchers first looked at oncogenes like CCNE1 and MYC, calibrating the guide RNA so it activates only when these are massively amplified. Second, they targeted specific in-frame deletions in lung cancer. The system spotted these unique junctions and wiped out only the mutant cells, leaving the healthy wild-type cells completely unharmed.
To show just how ridiculously potent this system is, researchers measured the GR50. That's the concentration needed to inhibit cell growth by 50%. When targeting that specific EGFR lung cancer mutation we just mentioned, the GR50 was an incredibly low 0.055 nanomolar. That is astonishingly potent. It proves the system isn't just highly specific, it's a wildly efficient inhibitor of mutant cell growth.
Section 4, the P53 Holy Grail. Which brings us right back to that massive 40 to 50% problem we talked about earlier.
The biggest challenge with P53 is that most of its mutations aren't massive, easily identifiable deletions. They are single nucleotide variants, or SNVs. We are talking about literally a single letter change in the entire genetic code. Building a chemical drug to distinguish a protein with a one-letter difference is a monumental, almost impossible challenge for traditional medicine. But for an RNA-guided nuclease, it's just a matter of precise base pairing.
Now, what's really interesting about this slide is the elegant trick scientists used here to turn the cancer's own mutation against it. Cas12a2 requires a specific sequence next to its target to activate. It's called the protospacer flanking site, or PFS, and it needs to be rich in the nucleotide adenine. Well, it turns out that some of the most notorious P53 hotspot mutations, like R248Q and R280K, are caused by a G to A substitution. So, the cancer's own single-letter mutation literally creates the exact adenine-rich trigger that Cas12a2 needs to turn on the shredder. The researchers effectively hijacked the mutation to guarantee activation. It is brilliant.
Looking at clinical data across nearly 17,000 patient samples, the researchers found something huge. Over 25% of all P53 mutations create exactly this kind of new sequence that Cas12a2 could easily target. That is an enormous real-world application. A single technological approach could theoretically be programmed to tackle a quarter of the hardest-to-treat cancer mutations in humans.
Let's move to and see how this builds from isolated cells to living organisms. Section 5. Shrinking tumors in vivo. You can have the most potent cellular assassin in the world, but honestly, it doesn't matter if you can't deliver it to the right place. So the researchers packaged the genetic instructions for the system, the Cas12a2, mRNA, and the specific guide RNA into lipid nanoparticles, or LNPs. These are essentially tiny fat bubbles, very similar to the delivery tech used in recent mRNA vaccines, and they allow the therapeutic payload to be safely transported right into the cells of living mouse models.
The results in living animals were just striking. When they looked at mice with MYC-induced liver cancer, those that received a simple PBS control had a high percentage of their liver covered in tumors. But the mice treated with the MYC-targeting LNPs showed a significantly reduced tumor burden. The data directly confirms that this transcript-activated chromatin shredding actually works in a living, breathing model. They pushed even further with lung cancer models, using specialized LNPs to deliver the treatment. The result? Delayed metastasis and no obvious toxicity in the healthy organs. Now, maintaining scientific impartiality here, the authors did note a hurdle. Some tumors actually reduce their target RNA levels to evade the shredder. It's definitely a challenge, but one that is potentially solved by programming the system to target multiple transcripts at once. We've covered a lot of complex molecular ground today, but it all culminates in this direct quote from the paper. Transcript-activated chromatin shredding provides a new approach to precision disease treatments for undruggable targets. This isn't just a slight, incremental improvement on existing drugs. It's a foundational shift in how we might treat the most persistent, difficult cancers known to medicine. We are standing at the absolute frontier of precision medicine right now. Think about it. If we can train a molecular machine to hunt down a single mutated letter in a vast sea of genetic code and shred only the cells that harbor it, what else is possible? What other undruggable diseases could we completely eliminate by shredding their genetic instructions? Thank you so much for joining me for this explainer, and I hope this leaves you as fascinated by the future of biology as I am.